Hello, I'm Sun Yan. Due to visa issues, I can't make it to FG this year. Today I will be presenting my work exploring mental prototypes by an efficient interdisciplinary approach, interactive microbial genetic algorithm. Nowadays, facial expression-based techniques have flooded our daily lives. For instance, facial expression manipulation techniques can be found in social media, and facial expression recognition techniques are used in social robots. Well, in our work, we deal with two challenges. For the first challenge, most applications are based on the 6 so-called basic emotions of Paul Ekman. However, as a cognitive reference point for the category, the universality of Ekman's prototypes is now being challenged by a growing number of psychologists. For one emotion, there should be many prototypes. For the second challenge, most facial expression-based systems can only deal with 6 basic emotions, whereas there are already more than 4,000 emotional labels. This is not enough to meet the needs in real life. The root of these challenges is the database. Because most existing databases only have the labels of basic emotions. A very small number of databases contain limited non-basic emotions, yet, creating such a reliable and labeled database requires expert knowledge, and it is time-consuming and labor-intensive for human annotations. Considering the aforementioned challenges, to address more critical areas such as stress detection, psychotherapy, and human-computer interaction, a system should fulfill the following requirements. First, the system should deal with more emotions. These emotions are not limited to basic emotions. It should include emotions that are not explicitly available in existing databases. Second, for each emotion, the system should provide multiple emotional prototypes. Third, the entire process should not require expert knowledge. The last, the system should take user fatigue into account, which means no longer than 15 minutes. Here, we propose our interactive microbial genetic algorithm to meet all the requirements. To deal with more emotions, we get inspired by the psychophysical reverse correlation process, typically employed in psychology. And to meet the following requirements, we adopt the genetic algorithm. Here, by an example extracting prototypes of anger, we demonstrate how IMGA works. The entire framework is a closed loop composed of the same 4 components of the traditional genetic algorithm: population generation, selection, crossover, and mutation. For initialization, based on the face of the actor, and by editing action units, we used a generative adversarial network to randomly generate 20 facial expressions. Then, we integrated the perceptual experiment of the reverse correlation. All 20 manipulated faces were shuffled and randomly regrouped into 10 pairs of faces, then the observer performed the perceptual experiment. In each trial of the perceptual experiment, the observer saw a pair of faces and was asked to choose which face best corresponds to the target expression. After collecting the answers, the corresponding action unit vectors of each pair of faces are annotated by winner and loser. Then the uniform infection and bit mutation operators are identical to the original literature on the microbial genetic algorithm. At the end of the current iteration, we updated the action unit vectors, thus regenerating the next generation of the population. When the process started iterating, we need to think about how to evaluate the system and when to stop. Differing from the traditional genetic algorithm, the fitness function is the subjective judgment of the observer. Thus, with limited trials in one iteration, we add the population evaluation module to evaluate the entire system and monitor the convergence of the system. Here, we compute the similarity between the current set of winners and losers, and the similarity between the previous set and the current set. Simultaneously activating too many action units brings visual artifacts that may influence the observer's judgments. That is why we add a 3-state constraint automaton to limit the number of activated action units during the crossover part. The dashed lines indicate that when the similarity computed by the population evaluation module reached the threshold, the system allowed each individual to have more action units activated, and when this similarity exceeded the threshold for the 3rd time, the system stopped. "12 observers joined in the perceptual experiment. Each observer performed 4 perceptual experiments for 4 emotions, 3 basic and 1 non-basic. We collected all representative prototypes of these observers, and we compare them with state-of-the-art prototypes. According to the evaluation, we can answer the following 4 questions. First, are our IMGA-generated prototypes compatible with state-of-the-art prototypes? The answer is yes. We can find out that all representative prototypes generally convey similar emotional expressions. These figures show the proportion of each action unit that appears in our representative prototypes. Most action units from state-of-the-art prototypes can be found. Second, is there universality? The answer is yes and no. We can say, for the basic emotion, only some action units reveal universality while the prototypes don't have universality. When we look at the figure of confidence, the proportions of action units are not as prominent as those in the basic emotions. This can also answer the third question. Is there a discrepancy between basic emotions and confidence? The answer might be yes. In terms of the number of activated AUs, we can find out that most basic emotion prototypes have at least 3 AUs activated. For confidence, about half of the representative prototypes have less than 3 AUs activated. The last question, are mental prototypes diverse? The answer is yes. For each observer, IMGA can extract more than 2 prototypes. This indicates the diversity within observers. And only a small proportion of prototypes are coexisted between different observers, that is to say, diversity between observers. We also employ subjective evaluation. We asked the same 12 observers to select, for each emotion, the 5 happiest, saddest, angriest, or most confident prototypes respectively. We applied 2 measurements to observers' answers. First, the proportion of observers who still choose at least one representative prototype of theirs. We found out that most observers still selected at least one of their representative prototypes. Thus, representative prototypes can reflect observers' mental images. For the second measurement, we counted how many times each prototype was selected and ranked them. And we found out that the top 5 are representative prototypes and state-of-the-art prototypes are less preferred. To discuss the efficiency of our approach, we compared our approach with related works using reverse correlation. It turns out that only our approach can extract multiple prototypes but with fewer trials. On average, it takes only 10.8 minutes. To meet all the requirements, we propose a novel interdisciplinary approach by integrating reverse correlation process into an interactive genetic algorithm. Moreover, we add a population evaluation module to evaluate the entire population, and we also add a 3-state constraint automaton to incrementally increase the number of activated action units and determine when to stop. This work is supported by ANR Reflect and Randstad Group. If you have any questions about this work, please contact us. Thank you.